As we've talked about previously, gene regulation is really important that it occurs properly because when it breaks down, particularly in the cell cycle, it can lead to cancers. So remember, cancer is improper growth and cell division. Cancer can involve altered gene expression, and this can happen at any of those levels of gene regulation that we've previously talked about. So we'll look at a few examples of this and talk about how understanding gene expression could lead to better drug design. So let's look at P53 again. P53 was a tumor suppressor gene, and because of its importance in the cell cycle, we said that we found mutations in P53 in 50% of all cancers. So P53 itself is actually regulating the expression of many genes. That's because it's a transcription factor, as we've talked about. So here you can see it's involved in growth arrest. P53 increases P21 production, which inhibits cyclin-dependent kinases, and those block cell division or progression through the cell cycle. It also plays a critical role in apoptosis. It increases production of BAX, which increases cyto-C and caspase 9, which promote apoptosis. So you can see if you break this, you break apoptosis here. And if you break P53 here, you don't have these blocks on the cell cycle. Super important, right? But it's doing this by regulating other genes that are there. So one of the things that we're interested in is how are tumors different in terms of what genes they express than normal cells. This is really important because if we understand those genes that are being altered, we can design drugs that can counteract that. So let's look at this diagram here. This diagram is called a heat map, and what we see is each little line here is a specific gene, and we're looking at the expression level of it. Red is genes that are being expressed, and blue are genes that are being repressed or not expressed. So in pink here, these 1, 2, 3, 4, 5 different lines, these are different tumor cells here. In green, we have 1, 2, 3, 4, 5 different normal cell types. So what we want to do is we want to look at the tumor cells and the normal cells and compare them and look for differences in gene expression. So right off the bat, I think you can tell that there's this big block of genes that are being repressed in these three tumor lineages that are normally expressed in the growing cells. Whereas all these genes here are being repressed in normal cells, but they've all been activated or are being expressed in tumors. That's bad news, right? Maybe these are involved in the cell cycle, and the cell cycle is now on constantly. So this is an example of transcriptomics, where we're looking at all of the transcripts that are being made in there and looking for patterns there. Now, you can see here, right, that this is not really like easily comprehensible by humans. We can see the broad patterns, but you know what would be really good at understanding this? A computer. Computer algorithms, machine learning algorithms, things like this. They could find patterns in here and actually quantify it. So again, I want to give my heartfelt pitch here. If you want to do anything in life that involves science, it's really good to know some math and to know about computers so that you can, even if you're not designing the algorithms, you can at least speak to the people that can design the algorithms and you can talk the same language. It's really important to know a broad subset of skills, not just biology, right? You want to know math, computers, physics, chemistry, all of these things so that you have the ability to interact with other people who understand these things at a very deep level. So how can we use this? Well, we see like post-translational modifications being really important. Phosphorylation of cyclin B causes progression through the cell cycle. So in some cases, in tumors, we see phosphorylation happening when it shouldn't. We never shut this phosphorylation off, which leads to constant growth and division. Well, what if we could design a drug that could come in and block phosphorylation of this? Well, that would stop the tumor from growing. So understanding the specific mechanisms of gene regulation that have broken can allow us to design drugs to target those specific processes. All right, let's finish up with some review. P53 tumor suppressor has large roles on gene expression because it's what? Pause the video, think about that. P53 influences a large number of genes' expression because it is a transcription factor. All right. So we looked at a couple specific examples of gene regulation being altered, leading to cancer. So cancer is improper growth and division of cells. And cancer cells have large changes in gene expression. We saw that P53 tumor suppressor is often not expressed in many cancer cells. P53 is a transcription factor that normally controls expression of many genes regulating the cell cycle. When you break it, those genes are no longer regulated properly. They kind of go out of control. We also saw post-translational modification of cyclins can lead to them being constantly on. So you're not actually changing the sequence of the thing, but you're constantly putting a phosphate on there, and that's leading to cells that are always dividing. We could develop a drug that could target that, block that process from occurring, hopefully stopping the tumor from growing. All right, so these are just a few examples of how gene expression are important. So it's not just about the sequence of the genome, but where and when the genes are expressed. That's really important and interesting. Okay, that's it for chapter 16.